vestibular functional tests for Meniere's, because I'm going to cover a little bit of that today as well. And I also just want to talk, primarily this talk today is really about the research that we've covered in 2018. So in the last couple of years, we've had a bumper couple of years in terms of publications and new studies that we've started to conduct and have completed and published the results of. So they're the sorts of things I'm going to cover today. So this year, over the last year and a half, we've grown the lab team. We've included a newer member, which is Dr. Miriam Jackson, who's a molecular biologist, and I'll talk about some of the research she's been performing in the laboratory. We've also had Mr. Chris Pastras, oh, there is the mouse there. Mr. Chris Pastras has finished his PhD studies this year, he's been a great benefit to the laboratory over the last 7 years, or 6 years. He started out as a research assistant in the laboratory for a year and then went on to conduct a PhD, and I'll talk about some of his results from his research which have been published this year as well. We've also had Dr. Liliana Sokolik, who has been a research assistant in the laboratory for the last year and a half. She actually came from being a research assistant in Professor Ian Kurthwaiter's laboratory, so she's got a wealth of knowledge in terms of handling the animal research that goes on. And of course, we've got clinical collaborations with Prof. Gibson and Payal, which they both talked about. And fortunately, we've had Ian Kurthwaiter in the laboratory for a number of years. He's been probably a tour de force for supervising the sort of research that we've had in the laboratory, and has been essentially a wealth of knowledge and a guiding force for the sorts of research that we do as a team. He effectively brings together the whole Meniere's research team as a whole, so he's invaluable for that perspective. And of course, we're still based at the Brain and Mind Centre in Camperdown. We have increased our laboratory space over the last year and a half, and we have all the resources there that we need for the various forms of research that we have. So as I mentioned, this year in particular has been a bumper year. We've had 5 publications published. They've covered various things such as looking at our basic understanding of the peripheral vestibular system. And that's important from the viewpoint of understanding clinical vestibular tests. So the sorts of things that Luke talked about that are performed in the clinic and at the forefront of clinical treatment of Meniere's disease. They're great tools for the clinician, but they often come with a lack of understanding about what those vestibular functional tests actually represent in the periphery. So much of Chris Pastras's research has actually been focused on providing a better understanding of what those vestibular functional tests actually mean and why they indeed may be different in Meniere's disease sufferers. We've then also worked on new animal models of Meniere's disease, and I'll talk about those results specifically today as well. We've begun working on inner ear pharmacokinetics. What I mean by that is we've been working towards helping us understand when we apply drugs to the middle ear, so when you go to a clinician, often they might give you a gentamicin or a steroid injection through your eardrum. We need to know exactly where those drugs go, because essentially you can just putting the drug into a bony middle ear cavity doesn't necessarily mean that drug is going to make it to the cells that need to be targeted in the inner ear. And indeed, over the last 5 years, a number of researchers, particularly Alex Salt, who we work with in the States, have highlighted that drugs don't diffuse very well throughout the inner ear. So all of these steroid injections and various treatments, chemical treatments for Meniere's disease and other inner ear disorders actually probably aren't very effective at all, not because the drug wasn't effective, but because the drug didn't actually get to the inner ear. So we need to now develop new types of drugs or new techniques that get the drugs to the parts of the inner ear that they need to get to. And so we've started working on inner ear pharmacokinetic research as well. We've started developing new treatments for Meniere's disease, which is, I think many of you would probably be pleased to hear. I'm not talking about great new chemical drugs or anything. It's essentially low-level treatments, but low-level treatment strategies for enabling us to get the drugs to better disperse throughout the inner ear. We've started to look at the biology of the lymphatic sac. So Payal talked about the clinical study where she and Adrian Mori have been taking out tissues from Meniere's disease sufferers' ears during ablative surgeries and looking at them histologically. We've started to, and she also mentioned the fact that we'd like to be able to look at the endolymphatic sac in humans, but that's actually really hard to do. That's where we come in as animal researchers. We can now take out the endolymphatic sac from animals, specifically animals that have been treated in certain ways to mimic certain features of Meniere's disease, and then look at the endolymphatic sac of guinea pigs to help us understand exactly what's going on at the cellular level in Meniere's disease ears. And then, of course, we've also started to develop some clinical research projects, such as the viral study that Prof just talked about. So on to, initially I wanted to talk to you and introduce Chris Pastras. As I said, he submitted his PhD in early October, and he's been working on furthering our understanding of exactly how the vestibular system functions. So when you go to get diagnosed for Meniere's disease these days, if you're seeing a relatively good clinician who has got a wealth of experience for specifically diagnosing Meniere's disease, they will perform a number of vestibular functional tests like Luke presented today. And some of those include the video heat impulse test and the vestibular myogenic potential testing as well. But we don't have a great, and they're reflex responses. So they're responses of the eyeballs or the muscles that control the position of the eyes or the neck. So they're reflex responses. They don't truly represent what's actually going on in the ear. So if we see a change in those reflex responses, we're really kind of left to theorize about what could be underlying those changes in the reflex at the cellular level in the inner ear of Meniere sufferers. So Chris has basically been, one of the advantages of using experimental animals is that unlike humans, in guinea pigs, we can actually surgically get into their inner ears, and in the case of Chris, what he's been doing, he's actually been removing their cochleas surgically, which allows him to get direct access to the vestibular end organs in a living guinea pig, which is heavily sedated, heavily anaesthetized, and will not wake up. But this surgical access provides us a way to actually measure the function of the vestibular system in these animals whilst we perform various pharmacological or experimental manipulations to better our understanding of what goes on in the vestibular system. He actually started out his PhD studies actually trying to better understanding the role of the vestibular efferent system. And this is I won't talk about what the vestibular efferent system is specifically, but it's a part of the inner ear that we have really no good understanding of what it does, other than to say that it does something extremely important in the inner ear. It's the brain sending neural signals down to the inner ear to control its sensitivity. That's theoretically what it could do, but we just don't know if it does anything at all. So what Chris started out his PhD studies doing was to actually electrically stimulate the vestibular efferents in the brain stem of anaesthetized animals, whilst in the periphery measuring the functional responses of the cells and the nerves in guinea pigs' ears. And what he found was essentially he was very clearly manipulate the sensitivity of the peripheral vestibular system through this electrical stimulation. It was a great study, provided with a wealth of research, and helped us better understand exactly how the vestibular system responds to bone conducted vibration and air conducted sound, but ultimately didn't provide him with the result that he wanted. But it was a good study nonetheless. And I'm hand waving there because it's not specifically about Meniere's disease. He then turned his focus more towards trying to better understand what endolymphatic hydrops, which is the bloating of the membranous labyrinth that Prof talked about, actually does to the vestibular system. So Prof talked about, and when Prof does electrocochleography in Meniere's disease, he sees an abnormal functional response, this abnormal nerve response and hair cell response that he records with a wire through the eardrum. Likewise, there's theories about what bloating of the membranous labyrinth does to the vestibular system to result in the abnormal vestibular functional results that Luke touched on today. And so by directly measuring the functional responses of the vestibular hair cells in anaesthetized guinea pigs, whilst performing various experimental manipulations that mimic endolymphatic hydrops, Chris was able to demonstrate that indeed you can increase or decrease or even increase the sensitivity of the vestibular system simply by hydrostatically displacing the vestibular epithelium. Now that's, if I can summarise what I'm trying to say there, is that he provided the formal basis or evidence to say that endolymphatic hydrops truly can by pressure, manipulate the sensitivity of the vestibular system. That sounds like a simple statement, and to Meniere's researchers across the world, we probably all would have said, oh yeah, that was always the theory, we always knew that. But there was never any evidence to suggest that that was the case. So no one ever had any evidence to say that hydrops per se, and the increased volume of the membranous labyrinth, can in any way affect the vestibular system. Chris has now been the first to provide that evidence, backing up all of those decades worth of theories. So it's very important from that perspective. His research is really important, but it'll be glazed over because everyone already thought that that was the case, and they thought that they already knew that, which I think is very disappointing for Chris. But he's nonetheless had really great success with his animal research. And now on to the animal models of Meniere's disease that we've been working on for the last couple of years. I presented some of this research at the last year's symposium, where we're basically halfway through gathering the data. We've now finished gathering all the data, we've analysed the results, we've published the paper, and it was published halfway through this year. And moreover, I want to talk about some of the other things that we're doing with these animals as well. So my animal research really is focused on endolymphatic hydrops, trying to understand and develop a theory about how endolymphatic hydrops can develop in animals, how we can diagnose it, and how we can treat it. So Prof talked about some of the ways that endolymphatic hydrops can be induced or can develop, and the theories of how endolymphatic hydrops develops. One being this potential blockage of the endolymphatic duct, resulting in a lack of fluid being absorbed in the endolymphatic sac when you have some sort of challenge in the inner ear, resulting in a bloating of the saccule and potentially the utricle, and certainly of the membranous labyrinth in the cochlea. You've also got, what that also implies that you have to have an increased secretion of fluid into the membranous labyrinth. Well, that requires, where does the fluid come from? It comes from blood vessels primarily that line the cochlear walls. So those blood vessels have to become more leaky, which we know happens when there is some sort of ionic or immune challenge within the inner ear. When you have increased their permeability, water potentially, and I'm hand waving here because no one's properly demonstrated this, but the theory is that when you have an immune or an ionic challenge within the inner ear, those blood vessels become more leaky, and we think that water flows into the membranous labyrinth, causing Reiser's membrane and the whole thing to swell. And that's the theory. So you have to have two things occurring to induce endolymphatic hydrops. One is an increased, well, you can have two things occurring. One is an increased production of the water in the cochlea, and one is a reduction in the absorption of the water in the endolymphatic sac due potentially to a blockage within the endolymphatic duct. Researchers there's several animal models out there for creating endolymphatic hydrops in Meniere's disease. The classic one, I'm not going to talk about today, is for researchers to go and get a guinea pig, surgically drill into the back of its skull. When it's under anaesthesia, it doesn't feel anything. It's a very small little drill, it's just a bit of bone. Guinea pigs are really robust. I mean, I've started to do these experiments thinking it was going to be really harsh. You anaesthetise the guinea pigs. It's a small slit, it's a small drill, you stitch them back up, they wake up, within 5 minutes they are fine. It's no different than many of the surgeries that we might undergo in our own lives. So it's not a particularly invasive or damaging type of surgery to these animals, but indeed extremely necessary for our understanding of Meniere's. But the sac ablation model of Meniere's disease in guinea pigs has been the mainstay of understanding how or the animal model of Meniere's disease. The problem with that model is that Meniere sufferers still have an endolymphatic sac. So it's not a true representation of a clinical model of a pathological entity. So other models have become enticing over the years. One such model is to put antigen, like parts of a bacteria or parts of a virus, into the inner ear of experimental animals. And lo and behold, when you put antigen into the inner ear, over the next couple of weeks, these animals start to develop endolymphatic hydrops. In some situations, they can develop a low-frequency hearing loss. And that's been demonstrated as a potential model that demonstrated that immune dysfunction can initiate endolymphatic hydrops. One of the problems with this simple model is that the endolymphatic hydrops in these animals tends to reduce or recover within about 4 weeks after putting the antigen into the inner ear of these animals. Now, there's been a couple of models where investigators have continued to pump the antigen into the inner ear, and they've said, oh, look, we can keep the lymphatic hydrops going if we just keep giving them injections of the antigen, but that's not really a good model for Meniere's disease sufferers. There's other theories about what could cause the antigen to persist, or at least the immune response to an antigen to persist within animals, and Stephen's got various theories that have supportive evidence for having a genetic predisposition that makes a person not be able to clear the antigen, or rather be hypersensitive to when there is an antigen present within the inner ear. But within experimental animals that have been developed in the laboratory, there's actually not much been done in that realm. One of the things I also noticed that hadn't been done or hadn't been understood, people have basically been ignorant of, is the idea that antigen, so far lots of Meniere's researchers had put antigen into the perilymphatic compartment of the inner ear and provided and looked exactly how the inner ear clears that antigen from the perilymphatic compartment. And that's not really telling the whole story, because Meniere's disease is a bloating of the membranous labyrinth, and this sort of swelling, temporary swelling, is only indicative of probably changes in the permeability of the vasculature system surrounding the endolymphatic compartment. All of these models where they put antigen into the perilymphatic compartment provide evidence that there is a temporary lymphatic hydrops. You have a gross, often that results in a gross cellular infiltration. Immune cells flood into the perilymphatic spaces to gobble up and process and remove the antigen so that things settle down after a couple of weeks. And certainly that's been used to help us study and understand the dynamics of the immune system within the inner ear. However, one of the things that people had glazed over was, what is the role of the endolymphatic compartment in the inner ear in terms of processing antigen? Now, within the endolymphatic space, there are many situations where you might have cellular debris break off and float around in this space. You might have antigen like viruses, or foreign antigen like viruses or bacteria potentially make their way into this space. And within this space, the fluid called an endolymph is really abnormal. It's got a very, very high concentration of potassium, which is lethal to most cells. So the one thing you don't want happening is immune cells getting into that space to process and get rid of antigen. So one of the things that we haven't clearly understood is exactly how does antigen within the endolymphatic space get removed from the inner ear. Now, Bill had this theory of longitudinal drainage for many, many years, and everyone sort of just glazed over because they basically thought, and even I must admit, even when I came to first work for the Meniere's research team, Bill Gibson's drainage theory, I looked at it and went, oh, that sounds nice, but it's a theory, and all theories are, there's a million and one theories, but they lack evidence, and people still say the drainage theory lacks evidence. Well, it's fortuitously, in terms of in the course of me doing this, this study where we've been placing antigen directly into the endolymphatic compartment of guinea pigs, it seems that we have now proven, gone some way to proving Bill Gibson's drainage theory. Now, what we basically have been doing in these animals is to inject antigen into the endolymphatic compartment of the animals, stitch them up, recover them for either a day, a week, or 6 weeks, and thereafter we have to euthanize them very rapidly and without any pain. Then we take out their temporal bones, and we have a look at the temporal bones histologically. And rather than actually do the standard technique, which is what people do when they want to look at tissues, which is cut up tissues and put them onto slides and put them under a light microscope and then look down a powerful microscope at that one thin slice. Instead, we look at the entire inner ear using this technique called light sheet fluorescence microscopy. And what that involves is taking the entire temporal bone, chemically treating it to make it transparent to light, and then what we can do is create a very, very thin sheet of laser light that only illuminates one very thin section of this transparent temporal bone. And when you're only illuminating one very thin section and you look at that cross-section through the whole organ, what you see is essentially very similar to what you might see on a histological slide. But the advantage of this is you can move the whole temporal bone through the entire light sheet and build up an entire 3D image of what is going on within the inner ear. And And that's very important for the inner ear because a really complex structure, histological sectioning does not work very well because it damages and morphs things like rhizos membrane. It creates lots and lots of artifacts and is very prone to researchers picking and choosing exactly what slide they want to use for their research output to provide support for their own theory. When you do things in 3D, you remove all of that biasing, that user bias. Anyway, on to the results. So, as I said, the project involved injecting antigen into the endolymphatic compartment of guinea pigs. But first we need to do the control experiment, which was to just inject artificial endolymph into the endolymphatic compartment. So we're not injecting antigen, but we just have to go through that control step of just say, what if we just inject fluid first off? And what we see is that hardly anything changes within the inner ear. So here's the cochlea, and we've been primarily focusing on the cochlea and the endolymphatic duct and the endolymphatic sac of these guinea pigs. And when we inject artificial endolymph, we certainly don't see any evidence of endolymphatic hydrops, so I'm not sure how well this shows through. But this membrane here is what we're looking at in the cochlea called Reisner's membrane. And with endolymphatic hydrops, this would be displaced outwards. But in these guinea pigs, it's nice and taut, it's exactly where it should be, and there is no evidence of endolymphatic hydrops in these guinea pigs. Their endolymphatic sacs look really normal, and the endolymphatic ducts are their typical very, very narrow cells. So the control experiments demonstrate that the surgery and the experiment itself does not induce any unwanted outcomes. Then we moved on to putting the antigen in the perilymphatic space. Now, others have done this, and indeed the results we found pretty much align with what they find, is that we tend to see sometimes we see a mild displacement of the membranous labyrinth, but not much endolymphatic hydrops. We see cellular infiltrate in the perilymphatic spaces, and often we see a little bit of fibrosis in these ears, but not a lot going on after about a week, and certainly not much going on after 6 weeks. So these animals don't have chronic endolymphatic hydrops, even though they often do have evidence that immune cells are flooded into the inner ear to get rid of the antigen. We then stepped over to putting the antigen in the endolymphatic space. Now, I should point out that we actually dropped the concentration of endolymph that we'd put. So in the animals we put it into the perilymphatic space, we decided to put 10 times less of that concentration into the endolymphatic space to see what would happen. And that would result in 0.1% lipopolysaccharide, very, very small volume injections, 0.4 microliter injections, and then recovering the animals for a day, a week, or 6 weeks. And these are just representative examples. Lo and behold, we didn't expect things to be so severe. Now, you'll see here that basically once you put the antigen in the endolymphatic space, you get massive amounts of immune cells just flooding into the inner ear, destroying everything. These animals are functionally deaf, and they have huge endolymphatic hydrops, but they also have massive amounts of cellular infiltrate and even evidence of ossification throughout the inner ear. So these guys did not fare well, even though we assumed initially we were putting very, very, very small amounts of antigen into the endolymphatic compartment, much less than what we would put previously into the perilymphatic compartments. That demonstrates that the antigen, when antigen makes its way into the endolymphatic compartment, the immune response is much, much more severe. One of the other things we noticed as well was that the endolymphatic duct in these ears was not blocked up like we had assumed it might become, but indeed quite the opposite had happened, and that it was swollen. So the endolymphatic duct, when you have this very large endolymphatic hydrops, actually bloats and increases in volume. Now, there's a whole bunch of other results here. I'm skipping forward to say, okay, because we started to work down the concentration until such level time as that we didn't have this huge influx of immune cells. We basically ended up with a result that with a 20-fold reduction in concentration of antigen applied to the endolymphatic compartment, we now have the situation where we can induce severe endolymphatic hydrops in these animals after 6 weeks with very minimal to no cellular infiltration. That's the result that we were actually gunning for at the start, and it's the result that we got, because we wanted to have an animal model of chronic endolymphatic hydrops of at least 6 weeks with no fibrosis or cellular infiltration, because that's the sort of thing that we see in Meniere's disease sufferers. Meniere's sufferers don't have significant amounts of immune cell infiltration into the inner ears and ossification, etc., etc. For all intents and purposes, they just primarily have an increased volume of the end lymphatic compartment. Indeed, in some cases you do have a bit of cellular infiltration, but often that wasn't the case, and the end lymphatic hydrops does seem to increase from 1 day to 6 weeks. So there does seem to be an ongoing increase in the volume. The end lymphatic duct in these animals wasn't always the same. In some animals it was enlarged, in others it was its usual narrow self. One of the things that we did notice is in many cases, particularly within a day of putting the antigen in the end lymphatic compartment, the end lymphatic sac secretes this glycoprotein that blocks up the entire end lymphatic sac and most of the end lymphatic duct as well. And now that's important for our research, and others had already been known, but this provided yet another example of this sac secreting this substance into the end lymphatic duct. And we think that's really important. It's one of the things that we're starting to focus on as well. We then started, so not only did we have a look at those slices, we also did 3D volumetric analysis of the structures. Now, which is really nice. People that do slice histology can't do that. One of the disadvantages of having a really nice microscope that can take lots of samples to a structure is that you then have to go and actually manually process all of these volumes. And we ultimately had about 56 temporal bones, and you had to then go through and with 1,000 images per temporal bone, and in each temporal bone you had to go through and by hand draw out all of the different regions of the inner ear. And doing a 3D reconstruction of each of these compartments took about 3 hours each one. So that was a task that I wasn't going to do. I was able to actually get a summer research scholarship and student and say, here you go, here's a bunch of data on a computer. Your job's to sit in a dark room for 8 weeks and analyse it. But he did it, and he did a great job, and it provided us with the volumetric data that we needed to support the suggestion that, which was obvious in looking at the images, that the end lymphatic volume is increased after 6 weeks in the animals that have been treated with 0.01% lipopolysaccharide. The problem with this animal model, though, is that the hearing loss is still quite severe. So even with the lowest dose of antigen that we'd used, the animals still had a fairly severe hearing loss. Many of them didn't have any sort of cochlear function. And so even though that histologically it looked like it was a good model of Meniere's, functionally it wasn't a great model. So we're probably still looking at having to either drop the concentration down a little bit more or look at exactly where we're putting that antigen, I think. And so we're still working on it, but it provided us a good model for developing, having a look at the structures and the histology of what is going on in the ear. Now I'm going to jump there. Here's just go through this slide. I may just skip it. It's essentially more evidence of the end lymphatic sac and the end lymphatic duct secreting this glycoprotein into the structure. And that's one of the things that we've been really interested in, is exactly what is involved because the sac, as we know, is really important in terms of Meniere's disease. Its ability to secrete glycoproteins into the luminal opening of the duct is really fascinating. Where the glycoprotein is generated and secreted from isn't exactly clear, and what the cellular mechanics are of that process are also not clear. So one of the jobs of Miriam Jackson to do in our laboratory has been to actually take out the end lymphatic sacs of these animals and actually start to understand through quantitative PCR and molecular biology techniques what's changing in the tissues that line the end lymphatic sac that induce that secretion of glycoprotein into that lumen. Because we think if we can start to understand the cellular processes that are going on there, we may be able to develop new therapies that target that cellular secretion of the glycoprotein, because we actually think that some cases of Meniere's disease may actually be an abnormal production of that glycoprotein, and if we can dumb down those processes, we may be actually provide a new therapy for Meniere's disease. One of the other things that was interesting, but this idea that we're putting antigen in the ear was nice, but it's not all that simple. A much simpler process for getting this end lymphatic sac to secrete glycoprotein is a much simpler technique where you just give animals glycerol. Now, this has been demonstrated many years ago, where you give an animal glycerol via an intravenous line, the end lymphatic sac secretes glycoprotein, and then the glycoprotein fills the sac. There's theories that glycoprotein draws the endolymphatic fluid from the rest of the cochlea up into the sac so that it can be further absorbed. And so rather than putting antigen in every animal and then looking at the molecular biology of the endolymphatic sac, we decided to switch towards using using glycerol, intravenous injections of glycerol in animals to help us understand what is actually going on at the cellular level in these animal models when they're secreting glycoprotein. In doing that research, which is what's been driven by Miriam Jackson at the moment, one of the other things that occurred to us was that this idea that glycerol actually triggers the sac to secrete glycoprotein that then absorbs endolymph fluid from the cochlea may actually present us with a unique opportunity to actually increase the ability of drugs to perfuse throughout the inner ear. But as I mentioned before, that when we put drugs into the middle ear, so when a clinician puts gentamicin or steroids and they do these injections through your eardrum, that drug does not diffuse very well throughout the entire inner ear. It tends to just essentially sit on the round window. If it does get through the round window, it permeates very, very locally, and there's not a very large diffusion of that drug throughout the inner ear. But we know that as far as steroids are concerned, one of the areas that we want those steroids to be getting to is the endolymphatic sac. And indeed, there's been a number of surgeons over the last 2 years have actually taken the approach of when they're doing surgically accessing the endolymphatic sac in Meniere's patients and actually injecting steroids directly into the endolymphatic sac, and the outcomes of those studies have actually been quite positive. Unfortunately, doing sac surgery is not something you can do in your clinic. It requires surgery, it's quite invasive, and it's probably a technique that you don't want to undergo. If we could circumvent that approach by using glycerol to actually attract fluid towards the endolymphatic sac, potentially carrying the steroids from the middle ear with it, we may be actually able to get more steroids towards the sac and provide a better treatment. The nice thing about this is that clinically, many of these patients are already given steroids and they're already given glycerol. We just have to give them together. And that's like a no-brainer. Simple clinical technique to do, already FDA approved. The nice thing is, another additional fact of this is that the only other situation where people are given both steroids and glycerol together is in meningitis. So in cases of meningitis, patients often lose their hearing, and one of the therapies has actually been to give both steroids and glycerol together, and in those clinical studies, patients the treatment was safe, and actually they had better hearing outcomes. It wasn't understood why they might have better hearing outcomes, but we think that potentially this idea that glycerol is somehow changing the pharmacokinetics of the inner ear might have something to do with it. So we're now starting to focus on this. And it would be really advantageous as well, because steroid treatment for the inner ear for Meniere's disease is on the rise. Many of you would have heard about various studies that have been done by this company called Autonomy. They've essentially done several large international phase 3 clinical studies of dexamethasone as a therapy for Meniere's disease. The company was booming, and then all of a sudden the results of their first phase 3 clinical study came out saying that they didn't reach statistical significance, suggesting that their clinical treatment of putting dexamethasone onto the middle ear didn't work, and their share prices plummeted. And everyone went, it doesn't work, glycerol, steroids don't work. Oh well, move on to something else. The company actually had 2 other studies on the go, and those other studies have actually started to now demonstrate statistically significant effects, demonstrating that steroids do actually provide a benefit for Meniere's disease. It's a fairly routine clinical surgery, and certainly in Europe, a lot of surgeons are starting to head towards providing steroid injections as a means for treating Meniere's disease sufferers. Unfortunately, not all Meniere sufferers are the same. Some Meniere sufferers benefit greatly from steroid injections, and others, it doesn't do very much for them at all. I suspect that one of the reasons for that variability may actually be that individual sufferers have different pharmacokinetics. The drug's not going to diffuse the same in every person's ear. Some people have big ears, some people have thick membranes, some people have all sorts of different anatomical reasons why drugs will not diffuse as well in one person versus another, and that variability may be the reason why steroids don't work the same across all sufferers. So if we can either work on techniques that allow steroids to diffuse better throughout the inner ear, and what people are starting to look at currently as well in America, is to try to come up with different drug formulations of steroids that allow those steroids to actually diffuse better throughout the inner ear. Because steroids, by their nature, are either water permeable but don't last very long in tissues, or they're water impermeable, so they're gels, and they sit on the tissues for a long time, but they don't diffuse very well and they're not very effective. So we need to actually work on the pharmacokinetics of those drugs, and I think that's going to be a great avenue for the next 5 years for Meniere's therapy. But I wanted to discuss more this idea that glycerol can induce this flow of end limbs. The first evidence we had of this actually occurring was through a series of experiments where I was injecting a fluorescent marker into the endolymphatic space of guinea pigs, and happened to be giving them intravenous glycerol as well. And what I found was this huge uptake. Here's the cochlea of one animal. The fluorescent marker went all throughout the membranous labyrinth, but there was this huge uptake of the marker up into the endolymphatic sac. And I never ever saw this in animals where I didn't give them glycerol. And this was the first sign to me that there is absolutely something going on with the drainage of the endolymphatic sac post-glycerol therapy in these animals. Now this marker was, as I said, I was injecting it directly into the endolymphatic compartment. That's not a great mirror for putting steroids into Meniere's sufferers, because we don't put steroids into the endolymphatic compartment of humans. We're stuck with putting the steroids directly onto the round window, which is a very different compartment. So then I had to actually ask, well, okay, what happens if I put the fluorescent marker on the round window in the middle ear of these guinea pigs and then give them glycerol? Lo and behold, the same sort of effect was seen. Here's an animal where I have put the fluorescent marker on the round window. Again, it's taken up throughout the cochlea, but it doesn't, without glycerol, it doesn't really get taken up into the endolymphatic duct or endolymphatic sac. When I put the marker on the round window and give the animal glycerol, there's a greater diffusion of the fluorescent marker throughout the cochlea, and there's this large uptake of the marker into the endolymphatic duct and endolymphatic sac. So I'm sort of starting to further this idea that we can use glycerol to get steroids towards the endolymphatic sac. But this is a fluorescent marker. It's not a steroid, its pharmacokinetic properties are very different than what you might see from steroids. So then I had to step over to, okay, well, how am I going to look at steroid diffusion throughout the inner ear with glycerol? So I started using fluorescently tagged dexamethasone. So here's some preliminary results I have from guinea pigs, where I've got fluorescently labelled dexamethasone. I've applied it to the round window of these animals, and I've given them intravenous glycerol. Now in animals where I don't give them glycerol, here's where the steroid goes throughout the cochlea. It doesn't diffuse very well, which is sort of in line with what other people have suggested occurs with dexamethasone. And there's almost no dexamethasone coming up into the endolymphatic duct and endolymphatic sac. But when you give the animal glycerol, the steroid seems to diffuse better throughout the cochlea, and there is a bit of uptake of the steroid into the endolymphatic duct. Not much being taken up the endolymphatic sac in this, this animal was basically terminated about an hour after putting the steroid onto the round window. So I think if I'd left it for a bit longer, given it a bit of more glycerol or something, I could have actually seen a bit better diffusion of the steroid up into the endolymphatic sac. Now, this, I think, is a simple approach to providing a better therapeutic advantage for Meniere sufferers. It's certainly one that I know that there's a couple of clinicians have already tried and tested around the world, and have actually anecdotally suggested that their patients do better with the steroid injections. So they've already been giving their patients steroid injections. They started to add on get the patient to drink glycerol at the same time, and anecdotally they've said that actually those patients seem to be doing a lot better than just the steroid outcomes alone. But that's not a clinical study, and anecdotal evidence is worth nothing in the scientific community. As I mentioned, one of the other things we're looking at as far as the glycerol treatment is concerned is looking at the molecular biology of the endolymphatic sac. So what goes on in the cells of the sac to secrete glycoprotein or to help increase the absorption of fluid. The reason we want to look at those mechanisms is because we want to target therapies to actually manipulate the role of fluid absorption, fluid regulation in the endolymphatic sac, and we're only going to do that if we understand what's going on at the molecular level. Sounds technical. One of the biggest hurdles of that particular study has been surgically getting out these wincy, tiny little bit of floppy tissues from the end lymphatic sacs of guinea pigs. Payal talked about the difficulties she has in surgery of getting tissues out during labyrinthectomies in Meniere's patients. Guinea pig ears are about 10 times smaller, and the end lymphatic sac is a really small structure, and it's adhered to the dura mater and blood vessels and all these other things that you don't want when you're trying to look at tissue, specific tissue. So one of the biggest hurdles for this research has actually been to develop the surgical approach of taking out the end lymphatic sac of a guinea pig. Not the simplest thing to do, but certainly necessary if you want to actually study the end lymphatic sac. And so I've already mentioned this particular novel adjunct therapy of glycerol and steroids is something that I'm currently looking at and hoping to pursue moving forwards, not just in the laboratory, but also in the clinical setting as well. So we'd hopefully within the next year start up a clinical study to actually look at the outcomes of intratympanic dexamethasone injections combined with glycerol therapy as well. With that, I wanted to, here we go, yep, right on time. Thank you very much for coming today, and thank the Meniere's Research Fund for their ongoing support of the laboratory, and also want to thank to Garnet Pass and Roddy Williams Memorial Foundation as well, who have been a great supporter of the laboratory as well. Thank you very much.
Introduce Bruce Kirkpatrick, who has been the driving force behind MRFI for many years and really has helped us to establish